So we are embarking on our last lecture with plants, transportation in plants. We're going to be talking about the vascular tissues, which includes our xylem and phloem. So a little bit of introduction here. Remember, terrestrial plants had to overcome many challenges, and the two big ones were maintaining water and nutrient balance, and then finally structural support for upright growth. And so the vascular system solves these two challenges. It helps transport water, minerals, organic molecules, sugar, and then we also have secondary growth going on, so that plants can grow in width and provide structure so it can grow up to obtain more sunlight for photosynthesis. So let's talk about this transport mechanism. Plants don't have any muscle tissue or circulatory system. So they move water through the cell wall spaces between protoplast of cells. They can move water and other things through the plasmodesmata. Because remember, our cell wall is rigid and it's box-like. And so how in the world do things get through? Well, there are actually tunnels or openings in the cell wall, and we call them plasmodesmata. They can move things through the plasma membrane. We've talked about diffusion and osmosis. There's a lot of interconnecting, conducting elements throughout the plant where they can move things. And so here's the general way or route that water takes. It enters the roots, it goes through the xylem, and then some exits through the stomata as water vapor through the process of transpiration. So water can travel long distances in the xylem. We kind of have this push-in pressure of water that enters the roots. Remember, cohesion and adhesion are taking place. We've been talking about this in chemistry. And then we also have a pull-in force caused by transpiration, because all of these water molecules, they're sticking to each other through cohesion. So as one goes up, it's pulling the next water molecule up. And then it's also clinging to the sides of the xylem vessel, the tracheids, depending on where it is in the plant.
About the pushing and the pulling of water molecules.
So here's a diagram. Now, when I was in college, we had to there was chemical formulas. This usually means some kind of osmotic pressure or something, but it usually flows from low to high. Yes, water potential gradient. Yes. So yeah, it goes through the roots, and we have cohesion and adhesion in the xylem, and then we have transpiration. So everything's connected, pearls on a string, pulling it through, etc. This diagram does a better job showing that cohesion, how they're linked together through hydrogen bonds. This is from high to low. My bad. I was like, huh? No. High to low. Osmosis moves from high to low. My bad. Okay. Moving on. So we have water that can diffuse through the plasma membrane. However, charged ions and other organic compounds that are too large need protein transporters. Some of these protein transporters, they don't use energy, so we call it passive transport. It flows from high concentrations to low concentrations. Other protein transporters, sometimes you have to move things against the concentration gradient, so you are going from a low to high, so you're climbing up the hill, so you need energy, and so these require ATP. And then you can, predict which way water is going to move by looking at the concentration of solutes, so I'm going to go back to the slide. Because these values are lower compared to these, I did it again, didn't I? These values are higher compared to these values, then water will flow up towards the tree.
Makes sense.
Yep. So there's that crazy little pitchfork symbol. That means water potential. And so we have to look at the water potential of water to figure out which way it's going to flow. There's also other units called megapascals. I won't quiz you on megapascals or anything. Just know that how water moves depends on the water potential inside of the plant. Okay. All right, movement of water by osmosis. So once water enters the cell, it's obviously going to expand because it's going to the vacuole, it's being stored. And so when a cell expands, we have it's called turgor. So the scenario is, here I have a plant cell, I place it into water where the concentration of solutes inside the cell is greater. So if I were to draw this, Sorry for those that are watching the video lecture. Okay, so here is my beaker. All right, there's my plant. Okay, not bad. Concentration solutes inside the cell are greater. So I have high, I'll just put sodium, I don't know why, but high concentration of solutes inside. Well, because I have a high concentration of solutes inside, that means I have a low H2O. Does that make sense? Because there's more solutes than water, so there's a low. So that means water is high out here. So water always moves from high to low, so it's going to move from the environment inside the plant. The cell expands, becomes turgid.
Okay.
Questions on that? Because I think I give you another scenario here. Oh, here we go. Never mind. So in this diagram, it would be a hypotonic solution where the concentration's inside. So whatever, high solutes inside the cell. So that means there's low water inside the cell. There's high water outside. So it moves from high to low. So now our cell is turgid. Now, in animal cells don't have that cell wall. So if they take in too much water, they will lyse or they will explode. Okay, here's another scenario. Plant cell is placed into a solution with very high concentration of sucrose outside. So water leaves the cell. So I'm going to draw my diagram here. I'm going to draw a flower this time. It's a really bad flower. Okay. So high solutes. If I have high solutes outside, what's my water?
Low.
Low. So that means my plant has high water. So water's going to leave the plant to the environment because it's moving from high to low concentrations. So my cell membrane pulls away from the cell wall. The volume of my cell shrinks. We call this process plasmolysis. And so you see this right here. Okay, water potential and turgid pressure, how they all kind of work together, actually. We've seen plants that become wilted because they're losing water, and that's all because of the lower water potential and pressure and osmotic inside the plant cell. But then when we have water that enters the cell, pressure increases until it's turgid. How in the world do we calculate water potential? You have to look at two things. Physical forces, which means gravity. These are very, very small, so we don't really include it in our calculations. But just know that as our turgor pressure increases, pressure potential also increases. If there's no cell membrane, like a beaker, then we cannot have turgor pressure. So then these are just simple math problems here. If I just have plain old distilled water like in a beaker, it's 0, 0. But then when I look at pressure in solutes, that means S. It's -2, so then water's going to move inside. And then if it's even, it's going to be turgid or equilibrium.
Wow.
Yeah, it's, like I said, just be aware of it. Just make notes of it.
Okay.
If when you go off to college and you decide to be a botanist, then dive into it.
A botanist?
Yeah, or some kind of plant physiology class or something. Okay, but the second thing that they have to look at is the concentration of the solutes, and that's what that subscript S, solute potential, and that's the most important thing out of all of it. So if you just look at the solute concentration throughout the entire plant, you can figure out how water is going to flow. Is it going to leave the plant? Is it going to enter the plant? Okay, that's all I want to say about that. So here's just a diagram depicting all of this, again, with the concentration of solutes inside the cell. I actually made these diagrams because that's definitely smart notebook material. But you can see that I have lots of solutes inside the cell, low solutes outside of the cell. So my solute potential out here is lower than it is higher. So that means I have low water inside the cell, right?
Mm-hmm.
And high water outside.
So the water's going to move in.
So it's water's going to move in.
Move out.
Yep. Oh, I don't know about the solutes. Solids don't really,
They just stay.
they usually stay, or maybe they'll go through a transport chain.
So that's why it expands, because you're just adding.
Mm-hmm.
Otherwise it'd
Did
just
we
stay
do a
on
lab
full.
on this when we did this year?
Yeah, I remember talking about it at least.
In biology, we did like potatoes, and then we did starch and iodine.
Oh, I remember that.
Did we do one? Yeah, we did one in biology. Yes, we did. Yes, a long time ago.
Yeah, because I had to
Too
make
long
up.
ago.
Too long
That's it.
ago. Okay, another diagram just showing math and how there's either, if it's equal, there's going to be equilibrium, so no net force, and you can compare that to a plant. Okay. Now, moving on to something called aquaporins. Right. So we know that plants can move through in osmosis, from high concentrations to low concentrations, but there are specific channels in which only water can travel through. So scientists believe that water moved across the plasma membrane only through osmosis. Okay, that should be like only through osmosis. I don't know what I was thinking there. But when they looked at this, the rate of osmosis that occurred was much faster than they predicted. They looked at surface area and just the rate of diffusion in general. And they're like, well, water is moving in and out of the cell at rates faster than we thought they should be. So what's going on here? So they started to study the surface, and they actually realized that there are specific channels just for water, and they are called aquaporins. And aquaporins are found in animals and plants. What aquaporins do is they just speed up osmosis. They don't change the direction. They follow the gradient. It moves from high concentrations to low concentrations, and they're also extremely important for water balance and moving water into the xylem. And so here's what the aquaporin looks like. And so we've talked about beta and alpha helixes in our protein structure. So it's made up of some helices here. But the unique thing is it's got this positive charge down here. And water is polar. So it means it's got a partial positive end and a partial negative end. And so what happens is water goes through here, all right, and it uses forces of attraction to kind of move it in quickly and to kind of shoot it out. So I just thought that was really cool. Okay.
And the guy won a Nobel Peace Prize for that?
Yes, he did.
Impressive. I want a Nobel Peace Prize. Put that on the resume.
Invent something.
No, you don't get it for inventing stuff, so it's gotta be.
Okay, so water potential gradient is extremely important in order for water to transport from the roots to the shoots. And we know that the roots are the entry points. And it can only move water from the soil if the water potential of the soil is greater than the roots. So this is why if you lay down too much fertilizer or if there's drought conditions, water isn't going to enter the plant. Because you got to look at that solute concentration. And then once it gets inside the plant, then the plant actually does a really good job trying to maintain gradients, so that way it always travels from low to high concentrations. They will move sugar throughout the plant to make sure that the gradients are lower as you go up in elevation, so water will move there. Does that make sense? Or higher, I should say, higher in elevation, because then that means there's low water. So again, another diagram showing high water potential. Notice that these values are higher than these values. So water will move from low concentrations to high concentrations. And like I said, the plant will do a very good job moving sugars to make sure that this gradient goes from largest to least. Sorry, largest to least. That's not the word. Greatest to least. There we go. Those are the two words you want to use. Not only do fertilizer and solutes inside the soil have an effect, but we have to look at humidity as well. So transpiration can have a huge factor in water potential. Okay, water and mineral absorption. So it comes through the root hairs to enter the plant, and those root hairs again increase surface area. These cells are usually always turgid. And these root hairs also accumulate a lot of ions. So they use ATP in order to do this. And the reason for that is so that the water potential is always lower inside the roots than it is in the soil. So water will always come from the soil to the roots. There are 3 transport routes inside the cell. The first one's the Apoplast root. where it goes through the cell walls and the space between cells. We have the symplast route, which means it goes through the cytoplasm between cells. So usually plasmodesmata is kind of how I like to think of it. Be a bigger announcement. And then finally the transmembrane route, where it goes between cells and across membranes of vascules within the cells. Now, they interchange between all three. They're not exclusive. They switch back and forth depending on benefits and situations at the time.
Is this all cells like plant and animals?
This is plant. This is plant.
That's a bold prediction.
Okay, so here's a diagram showing the three. Here's my apoplast, transmembrane route, yeah. Symplast goes through the plasmodesmata, and then the apoplastic route. Mm-hmm. Okay. So we were talking about cell layer tissues, endodermis in this case. Molecules will eventually reach the endodermis, and it's blocked by the Casparian strip. So then the molecules have to pass through the plasma membranes and the protoplast in order to reach the xylem. So I'm actually going to go back a slide once you write what you need to write and show you the Casparian strip. Okay, so here's my Casparian strip. So now it has to go through a certain route in order to get past it to the xylem, because the xylem is inside the Casparian strip. Okay. Xylem transport. There's always root pressure, even when transpiration is not occurring. So, like transpiration means that water vapor enters or leaves the plant, and it's usually caused by like heat of the day, etc. But root pressure often occurs at night. It accumulates ions, and then transpiration from the leaves is very low or absent because a lot of this takes place during the day. So we have ion concentration that builds up to generate that root pressure. So then water moves into the plant despite transpiration taking place. Because transpiration also helps move water, because all the water molecules are connected to each other, pearls on a string.
That's a cool picture.
Root pressure can also be so strong that water will ooze out at the tips. And so we call this cavitation or dew. So we've all seen dew before. But the root pressure is so great down here that it's pushing up all the water through the plant to the edge of the leaf, and then it comes out like dew. But just keep in mind, root pressure, again, helps, but transpiration is that main force of moving water.
I never knew it was like plants that were actually excreting water. I always thought dew was just like
on the outside.
The air
Yeah. No, it's from the plant.
Oh.
Okay, moving on to those vessels and tracheids. So remember, water has this tensile strength, all from that cohesion of molecules. And cohesion comes from those hydrogen bonds because water has unique properties due to those hydrogen bonds. So we have this cohesion tension theory, where tensile strength of the column varies inversely with the diameter of the column. So we're looking at the strength of water based on the diameter that it is in. So the smaller the diameter, in this case, like diameter of the vessel or the tracheid, the greater the tensile strength. And so you may remember from biology, the capillary lab. I think we did like paper clips in a cup, and then we did capillary tubes. And you notice that the diameter of the tubes were various lengths. And so the one with the smallest diameter had water go up, travel the furthest in it because of this greater tensile strength. So, okay, the effect of cavitation. So tensile strength does depend on that water column. So if you have air bubbles, then your column gets broken, and then cohesion fails. So we call this cavitation. And when that happens, there's no water transport. Plant becomes dehydrated, death to that part of the plant or all of the plant, depending on where it is. So that's not a good thing. Okay, rate of transpiration. Okay, so transpiration is when water vapor leaves the plant or water leaves the plant, and it usually exits through the stomata. So our stomata have the ability to open and close. They're made of 2 guard cells, and based on turgid pressure, they'll open and close. And they'll open when it needs carbon dioxide, but water vapor is also a byproduct of photosynthesis. 90% of the water that's taken in through the roots is lost to the atmosphere through transpiration. So there's some conflicting requirements because the plants need CO2, but they don't want to lose all of their water to the atmosphere. So how in the world do they do this? They use the stomata and the cuticle to help balance their needs and their loss. So there's that turgid pressure inside the guard cells that causes the stomata to open. So when the stomata is closed, there's low water in the vacuole. So you can kind of think of it as it's wilted. And then it will take in potassium and chlorine and another substance called malate 2 through active transport. Solute concentration increases, thus my water potential decreases, and then water will enter the cell and it will become turgid, and when it's turgid, that's when it opens. Okay. So here, my guard cells, they are dehydrated. Okay, they don't have a lot of water in them. They're flaccid, I think is the word that the book uses. So it's going to take in potassium and chlorine, and then as a result, water is going to rush in. Now those guard cells become turgid, and they open up, and then they lose the ions, so that way, or when they want to close, the ions will leave, and thus water will leave, and then they'll go limp and close. So another diagram, turgid, not turgid. Okay. Water stress responses. How in the world do plants adapt to drought conditions, or what strategies do they use in order to limit water loss? So the first one that we just talked about, stomata, and this is for immediate response. So they will regulate and open and close during key times of the day to prevent water loss. There are also morphological adaptations. So like loss of leaves to limit transpiration. So plants will drop their leaves during drought periods. Another adaptation is maybe the leaves will be thick, so they'll have like a lot of cuticle. Their leaves might seem hard, but these leaves will have few stomata. So with few stomata, then you can't really lose that much water. Another adaptation, leaves are covered with trichomes, which is those tiny little hairs, and the hairs actually reflect sunlight and reduce the heat load onto the leaf. And these hairs, they look white microscopically. I think I have a picture, shoots, but yeah. They can also respond to flooding, so like short-term hormonal changes and long-term adaptations. So when flooding occurs, oxygen in the soil decreases, and it will interfere with how sugars and minerals are moved. So we're going to see abnormal growth as a result, and hormone levels inside a plant change. And yes, plants do have hormones. So what they'll do is maybe they'll form a larger lenticelle, which is a structure inside the plant that is used for gas exchange. And so here you can see all those tiny little air spaces that they can store oxygen when it's submerged underwater. So we call it arenchyma. And arenchyma kind of sounds like air, but chyma means a type of tissue inside the cell. Phloem transport, sugar. So a lot of carbs made in the leaves are transported throughout the plant. So we call this process translocation. Now, a lot of kids have this misconception that plants, they only do photosynthesis, but they also use cellular respiration. So plants have the ability to do both. And so the sugar that they make in photosynthesis is actually used to build structures inside of the plant. So we have organic molecules are transported up and down the plants. Aphids, which are insects, when they pierce the phloem cells, sap continues to flow out of it. Another thing that we may not think about phloem, not only does it move sugar, but it can also move hormones that the plant makes. Turgor pressure differences also drive phloem transport because, why waste all the energy? Like, let's just use passive transport. So we call it the pressure flow theory.
What are we doing for review tomorrow? Are we playing Code or something?
No, I think I'm just going to whiteboard talk.
Okay.
And ask you guys questions. Okay, so I'm going to talk a little bit about this slide here. Sugar is formed in the leaves, so that's where our carb comes from. The carb source is from the photosynthetic leaves. And it has to travel down to the roots because, the roots have to grow and extend out, so that it uses carbs as one of its fundamental building blocks. And so we call food storage tissues or growing tips or maybe roots sinks, okay? So we kind of have this process where sugar is moved from the source to the sink. So we call this phloem loading. This is where the carbs or the sugars, they will enter the sieve tubes. Sieve tubes are vascular tissues, another kind of like sub tube underneath phloem. And it'll travel through, there's a few ways it can travel through. Apoplastic or move across the cell membrane. If it needs to use ATP to move, there's also companion cells and parenchyma cells that will provide ATP to move that. But a lot of times once it enters in the sieve tube, there's no energy because it's going to make sure that the gradient goes from, in this case, greatest to least. So we'll move from high to low. So a diagram showing this. So here's my xylem, again moving up or with the potential gradients. But sugar, it's made in the leaf, so it's higher up, so a lot of times it'll flow down. So here's my companion cells that help move the sugar and load it into the phloem, and then they just make sure that the gradients always go from least to, or make sure that sucrose travels from least to greatest. And then once it reaches the sink or the root cell that it needs to, it uses a companion cell again to kind of move it and utilize it to the cell that needs it. So companion cells help load and unload it. You can think of it as that way. There's also pressure flow with the sugar and water. So, water tends to move up through the xylem, but sometimes it has the ability to travel down through the phloem as well. So, okay, awesome. That's all I have.